we are going to learn is protein synthesis. You guys might have heard so much about it. This is another main aspect of DNA. So let's move on. So what are amino acids? Amino acids are basically monomers, meaning building blocks of proteins. Then we have base triplet. So base triplet is what? Three nitrogenous bases, one after the another on the DNA. After base triplet, we have codon. These are three nitrogenous bases, one after the another on mRNA, complementary to the triplet on the DNA. Then anticodon, these are three nitrogenous bases, one after the another on tRNA, thus are complementary to mRNA. We have already discussed about the functions of mRNA, tRNA, and rRNA. So you might go look back into it. So we have transcription. So transcription is the first stage. Then the other stage is translation. So don't confuse between those two. First we transcript the information and then we translate it. Do you get it? So yeah. Then first stage of protein synthesis. Then mRNA formed from DNA carrying the code for protein synthesis to be made. Then the translation, the second stage of protein synthesis, amino acid combine to form a protein. Okay. Then let's move. All right. So this is a structure of an amino acid, and you should know that they are linked to each other by peptide bond. So it's important, and they might ask you in this exam. So you should remember. This. All right. So three consecutive nitrogenous bases on DNA strand are called base triplet. Then three corresponding nitrogenous bases on mRNA are called a codon. Then the three corresponding nitrogenous bases on tRNA are called anticodon. So you should remember this. You should also remember that the bond here, it's the peptide bond in the amino acids. All right. Then let's move on to the transcription. So first you transcript the information and then you translate it. So transcription is the first stage of protein synthesis. This occurs in the nucleus here. Let's go through the process of it. Okay, here. So the process of protein synthesis. Protein synthesis take place in two steps, namely transcription and translation. All right. So transcription happens in the nucleus. So you should know that transcription happens in the nucleus. So what happens? DNA double helix unwind, and then the double-stranded DNA unzips, and the weak hydrogen bond between them break. One strand is used as a template to form mRNA. Using free RNA nucleotide from the nucleosome, the RNA is complementary to the DNA. mRNA has now coded the message for protein synthesis. Alright, so let me tell you one thing. So from here to here, it's the same process of DNA replication. The double helix unwind, the weak nitrogen bonds break. Instead of two strands, one strand is used as a template to form mRNA. Right, so then using free RNA nucleotides from the nucleosome, the RNA is complementary to the DNA, and now mRNA has coded the message for protein synthesis. Now let's go through the process of transcription. Okay, so the protein synthesis takes place in two steps mainly, which is transcription and translation. So transcription happens in the nucleus. So what basically happens is the DNA double helix unwinds, then the weak hydrogen bonds break, then the one strand is used as a template to form mRNA. Then using free RNA nucleotides from the nucleoplasm, the mRNA is complementary to the DNA and now mRNA has coded the message for protein synthesis. So from here, sorry about that. So from here to here, it's the same process of DNA replication. So this might be easier for you. Okay. So yeah, this is the process of that. And so let's, all right, so the double helix unwind, they basically unzip, the weak hydrogen bonds break, one strand is used as a template, then the RNA is complementary to the DNA, and now mRNA has copied the code for the protein synthesis. If you if you memorize this 5 steps, you'll also get your 7 mark. Okay, then let's move on also, because I remember in the exam what I did was I only memorized this 3 and this 2, meaning 5 points, and my teacher used to give me 7 mark. So yeah, and then it's very simple because it's the same process of DNA replication, there are only minor changes. Okay, so yeah, now let's move on to the translation. So translation is the second stage of protein synthesis. This occurs in the cytoplasm, this occurs in the nucleus. You should know the difference, sorry, don't mind that. And then this happens in the cytoplasm. All right, yeah. So yeah, here's the process. So each tRNA carries a specific amino acid. So when the anticodon on the DNA matches the codon on the mRNA, the tRNA brings the required amino acid to the ribosome, and then that amino acid becomes attached by the peptide bonds to form the required process. All right. So another thing you should know is to master this structure, because this will come in exam. Of course it would be a bit difference, but yeah, it would be like that. So let's go through it. Okay, wait, first let's go through the structure of transcription, then we'll go through that. Okay, so this inside part, it's the nucleus, then this is twisted, which means this is DNA. Then now mRNA transcription is happening here. Then now the mRNA has copied the code for protein synthesis here. All right, then if we go through here, these are the protein forming, then from the amino acids. These are the amino acid, then the tRNA bringing the amino acids, and this is the mRNA. All right, then the next thing we'll be learning is the mutuation. What is mutuation? Mutuation is a change in a DNA base sequence, all right, or a gene. So since mRNA is copied from the DNA molecule during transcription, this will result in change in codons. As a result, different tRNA molecules carrying different amino acids will be required. So the sequence of amino acid changes, resulting in the formation of different protein. So if the amino acid is coded for, there would be no change in the protein structure. All right, so in simple words, what this is trying to say is during the mutation, the DNA nitrogenous base sequence changes, which means that adenine might go with cytosine, then guanine might go with thymine. So this might happen. So when this change happens, so since mRNA copies the code for from the DNA, the mRNA would also change because there is change in codon. So therefore, all right, so if there is a change in mRNA, then there would be change in tRNA, because the mRNA copies the code for protein synthesis and takes it to tRNA. So if there is a change in mRNA, there would be change in tRNA also, and And then what would this result to? This will result to the sequence of amino acid changing, okay, resulting in the formation of different protein. So different type of protein would be formed. And if the same amino acid is coded for, there would be no change in protein structure. Let's attempt this practice question. So what I suggest always is you should pause the video, try to attempt the questions and then come and mark it. Alright, so let's start by reading this. What most of you usually do is so you don't read the headline and you simply move on to the question. But no, you shouldn't do that. You should read this question also. Alright, so it's saying that the diagram below is the systematic representation of protein synthesis. Alright, so this diagram is for protein synthesis. So 1.5.1 says that identify the process Z. So the process Z is transcription. Now so how do we know it's transcription? It's because you can see that double DNA double helix here, and then one strand acted as a template to form mRNA. All right. So as you can see that, so this process is basically transcription. And you see this little dotted line here. So that is a nucleus. So they might ask you in exam. So if they ask what's this, then you can say that it's nucleus. But yeah, the process is transcription. The next question is saying that identify the molecule R. All right. So molecule R is this one, right? So what's that? That is mRNA. How do you know? Because it copied the code for protein synthesis from the DNA. So that makes it, that makes it mRNA or messenger RNA. You can write whatever you wanted. Then organelle Q. Where is Q? Yeah, it's here, right? So that Q, it's ribosome. Now you'll ask me, how do you know that it's ribosome? Because tRNA brings required amino acids from ribosome to form the amino acids. Yeah, so it's because tRNA brings required amino acid to the ribosome, right? So this makes this ribosome. Give a collective name for nitrogenous bases O. All right. So here this is anticodon, because the nitrogenous bases on tRNA are known as anticodon. So you should know that. Alright, so as you can see here, the nitrogenous bases on DNA are known as base triplet. The nitrogenous bases on mRNA are known as codon, and then on tRNA are known as anticodon. Yeah, so this makes this a anticodon. Alright, then they say that they determine the sequence of nitrogenous bases at area S. Okay, here they're asking for here. Alright, so one thing you should know that in DNA, thymine goes with adenine and cytosine goes with guanine. Alright, so here it's thymine, so that would be A, cytosine, that would be G, and then again that would be A, so that would be T. So the base sequence is A, G, T. Okay, so yep, let's move forward. They say that which strand 1 or 2 was used as a template for the formation of molecule R. All right, so between 1 or 2, which strand is used as a template? So how do we identify that? Okay, so let's look here. This is a thymine. What would thymine change to? Adenine on the mRNA. and this is what adenine. So adenine changes to uracil on the mRNA. Remember that on the mRNA, thymine is replaced by a uracil. Okay. Then this is G, so that becomes C. Then this is also guanine, so this becomes, oh wait, it's twisted, right? So this is cytosine, so this becomes guanine. Then this becomes G also, and A becomes U. So it basically process 1, strand 1. I hope you understand that. Okay, so let me make it clear for you once again. On this DNA, A will always go with T. That's AT. But then on mRNA, it will go as AU. Okay, so you should know that. Then which amino acid 3, 4 or 5 will be brought to area P? Alright, where is area P? Oh, got it. It's here. So they are saying between 3, 4 and 5. Answer is 4. So okay, let me explain to you why it's 4. So here it's uracil. It changed with adenine. Then adenine changed with uracil here. And then guanine changed to cytosine here. Okay. Then yeah, then here CCA changed to GGU. So the only one left here is this one. So that would be here. So that would be number 5. Then they say that name the type of sugar that forms a part of structure molecule R. Alright, where is molecule R? Okay, it's here. So that is mRNA. So what type of sugar do we have in RNA? It's ribose sugar. So you write mRNA. And let me tell you one thing also. Here, you can see that this is a tRNA, right? So that would be U, A, and G. And then here you have A, U, and C. So you might ask me, okay, so of course uracil goes with adenine, but why didn't adenine go with thymine? Yeah, so why did adenine didn't go with thymine, but then go with, but then went with uracil? It's because only on DNA it goes with thymine, but then in mRNA and tRNA and rRNA, adenine always goes with uracil. So I hope this helps, and yeah, thank you so much for watching my video. And if you have any questions, you can just comment. I'll definitely try to answer it. And yeah, bye guys, and best of luck for your exams. Go ace it. You can do it. Bye.